All right, so we're going to prepare a DNA agarose gel for our gel electrophoresis. We're going to use 1%, and we're going to prepare 40 milliliters using the TAE. So first, we're going to weigh out our agarose. It comes out of this container, molecular grade, but I've weighed it out here to make it easier for weighing it out. So first thing we're going to do, we're going to come out here, we're going to tare, and we're going to add slowly just until we get to 0.4. That's too much, so if you put too much, you're going to have to start removing in order to get 0.4. So you just grab an extra one over here, and grab it. Start slowly shoveling it out, waiting for it to stabilize between goes. There is almost there, just a bit more. There you go, 0.4. So we're going to set that aside. We can turn off our scale, we don't need it. Next, we're going to measure out 40 milliliters of the 1X TAE buffer. So we're just going to pour some out here. You're also going to look at this horizontally, so you're going to go a little lower. It's just about 40 milliliters. And then we're going to add them all to this big flask over here. So we're going to go over to the microwave because we're going to have to microwave this. Let's come closer to our other agarose. Add our agarose here. We're going to add our TAE. Now it's at the bottom, we're just going to swirl it to do this. And now as you can see, it is not fully mixed. So what we're going to do is we're going to mix it inside the microwave. It's going to help it solidify. Just make sure any last bits are added. So I'm going to transfer this to a smaller flask here. Not only will it be easier for pouring, but it also did not fit in the microwave. We don't need this one. And as you can see here, it's just shy of 50 because we have about 40 in here. And as you can see, it's not mixed. So what we're going to do is we're going to heat it up to mix it and make sure everything is well dissolved. Now it's important to make sure you pay attention for the gel because it will spill out. So you're going to have to babysit this one. Every couple of seconds you're going to want to let it cool down. And you're going to want to make sure that it's not overflowing. So we're going to set it for 1 minute. We probably won't need the 1 minute, but just in case. All right, so we're also going to prepare our casting gel, our apparatus here. So this is unique in that it can clamp on and make a tight shape, has rubber on the sides to form to it. So how you use this one is you're going to take your chamber here, you're going to slide it in the middle. If you have two, you're going to put one on either side, but if it's one, you want to be straight in the middle. And as you can see, you can move this, it's kind of loose. What you want to do is you want to twist this so you hear a sound, and that will tell you that it's locked and it's ready to go. So when you pour your gel into here, it won't pour out, it won't spill out, and it will solidify. And what you hear is you have the combs, and there are little marks here. These kind of lock in, so it tells you exactly where you're going to put the comb. You can't mess this up, because once it locks in, it won't move. So we're gonna have this pre-prepared before we pour out our gel. As you can see, it's slowly bubbling. It's getting there. So we're gonna keep doing this until we see no more particulate left. So we're gonna let it cool down and sit for a second. Make sure it doesn't spill. We're gonna go 2 times here, and we're gonna see if that's enough. If not, we'll just add more time. Depending on how much gel you make, I see that it's bubbling, so I'm just gonna stop it here. It is getting quite hot, so I'm gonna use a paper towel to grab it. So you can see this probably needs a bit more time. It's getting there. There is some particulate left, so what I'm going to do is I'm going to add maybe 30 more seconds. It's bubbling. You don't want it to flow over, so keep watching the bubbles. If they get too aggressive, you're going to want to stop this. Be careful, it is hot. You don't want to grab it and burn yourself. As you can see, it's fairly clear. So that's a good sign for us that the gel is done and ready to go. So we're going to grab the gel. We're going to go to the cast that we made, and we're just going to pour it out. And we want to be very careful. If you have gloves, use them. I do recommend. You want to pour it out slowly as to not ensure there aren't bubbles. If there are bubbles, it's not the end of the world. We can deal with those. In this case, there weren't many bubbles. Then you're going to add your combs while it's still hot. Again, remember they do lock up. So fill around until you feel both sides have locked up and are ready to go. I don't know why it's not. Oh, there you go. It doesn't matter which side you do. And we're just going to pour out any remainder just in case. If you do have any bubbles, one tip you can do is you can use these pipette tips and just brush them off to the side. As long as they're towards the bottom or to the side, they don't impact your lanes, you should be fine. So as long as you're set, now you're going to wait about 20 to 30 minutes for it to cool and for the gel to set, and then you can use it for your gel electrophoresis. Okay, so after you've waited your 20 to 30 minutes, your gel should be solidified. So what you're going to do is you're going to loosen the lock on this. You're going to try to gently wiggle out your gel. Gently wiggle out your gel. You don't want to pull out the comb yet. Now that you're done, you don't need the casting apparatus anymore, and you can bring the well. As you'll notice, there's an arrow telling you which way the gel is going to run, so you want to run it that way. And once you put it into place, it will lock and it's ready to go. Then you're going to take your 1x TAE buffer, and you're just going to fill the well on both sides until the gel is fully submerged. So you're just going to make sure the wells are fully submerged and everything's submerged in your buffer. Just make sure you add every last drop. And now that you've done so, you're able to remove the combs. Again, you want to be gentle and you want to remove straight up. And if you've done it correctly, you should be able to see your wells. You don't need this anymore. And now you're ready to prepare your samples for loading. And now that we've finished our samples, again from the PCR machine, we're going to add eco dye, which is going to help visualize our DNA after our gel has finished running. So we're going to do the ladders first. So we're going to add 1.5 microliters of the eco dye to our samples. We're just going to grab the micropipetter, set to 1.5. Do that their second ladder. We're going to use 2 ladders for our experiment. And then we're good to go. We're going to add also to our 3 samples. We've added an extra sample of just control pretty much. And we're going to add 5 microliters. So we're going to grab a 5 microliter pipetter. Right over here, set to 5 microliters. And grab different tips. We're just going to add 5 microliters to all our samples here. We're gonna pipe it up and down to mix to make sure everything is good to go. You don't want to contaminate, so we're gonna switch pipette tips in between sessions. Again, 5 microliters, same as usual. Pipetting to mix. We'll mix it again right before we do it. And finally, our negative control. And now that we've done it, we're going to start loading our samples. Now that we've finished, we're going to make sure samples are in front of us, and we're going to start loading our samples onto our gel. We're going to add a ladder first, and then sample, 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 then finish off with another ladder. Most gels can load about 20, so we're going to try to load 20 microliters onto our samples. Although our ladder is less, we're just going to load 5. Let's start off with that ladder. Perfect. we're going to take one of our ladders. We're going to pipe it up and down, kind of just mix it. We're going to take the 5 microliters, and very carefully we're going to start putting this into one of our wells. You kind of want to go above perpendicular. You don't want to and then you're done. Don't go to second stop. You don't want to blow bubbles in. And then you just want to dispose of the pipette tip. Then we're going to load our samples. We're going to load our zero control, which we know what the sample is supposed to be. And this is here is where we might want to load 20 microliters. Take our zero control. We're going to pipette up and down just to make sure everything is good to go. And you want to kind of be perpendicular to it. You also don't want to poke through the gel, so you want to be kind of steady with it. Go. You just gotta do it for the last 2 samples. You should be good to run the gel. So again, pipette up and down to ensure it's mixed. Finally, you're going to load your last ladder. And that's going to be 5 microliters as well. And now that you've finished loading your samples, you're going to cover them up black to black, red to red, like always. You're going to hear some clicks once it's fully closed. You're going to collect it into your power supply. And we're going to run it at 90 volts, black to black, red to red. It doesn't have the wires are connected. You're going to put this to 90 volts. And once you're good to go, you're going to click run. And to confirm running, you're going to start seeing bubbles, which there are. And now you just wait about 45 minutes to an hour. So we're going to set a timer for 45. And we'll come back and check our results. All right, so now that it's finished, we're going to take the gel out, we're going to put it onto the imager. So we're going to be careful while removing the gel here. So we can pour out any excess, we don't need that. The gel kind of just slides off here. We want to be careful with the gel. I just put it in the center here. And now we're going to take it to the imager. Today we'll be talking about how to image your DNA gels using the ChemiDoc. dock isn't on, you're going to want to turn it on over here using this green button. And it's going to take a while to warm up, but when it's ready, it should look something like this. You want to click on the screen, and you want to sign in to the MMB lab. And when you sign in, it should look something like this. Before we image it, there's a couple of things we need to keep in mind. First, we need to make sure we're using the right imager and right tray. For DNA, you're going to need the blue tray over here, and you want to make sure it's the blue tray. If it's not, remove the tray and replace it with something, or replace it with the blue tray itself. You'll know you'll have it in the right position. You'll hear a little click, and the screen will acknowledge that it's ready to go, and you're going to want to push it in. The second thing we need to keep in mind is our application settings. So the first one is the type of application we're using. If we're using DNA, we'll click nucleic acid. If we're using protein, we'll click protein. And if we're using our Western blots, we'll click blot. In this case, we'll be imaging our DNA. So we'll click gel green DNA imager. And we'll click the X when we're done. The last setting we have to keep in mind is the exposure time. The exposure time depends on different settings. You have manual, which you'll manually set the exposure time. If it's your first time, I wouldn't use it. I would use the auto optimal exposure time. This will give you the best band quality and the highest definition. Once you're ready to go, you're going to open up your dock, pull it out, and then take your DNA gel. In this case, we're using a demonstration. You're going to center it as much as possible. Push your dock until you hear a click. Close it off, and we'll give you a little preview. You may or may not see your bands, and that's all right. We'll see them after we click image. So you want to click the green button when everything is ready. It's going to start imaging. And when you're ready, these are the results you're going to see. You're able to resize them, and you're able to analyze them as you want. All your results will be saved in the gallery, which you can then export using the export button to a USB in whatever format you want. Now, all this is a sample gel. Your gel should look something more similar to this. In this case, you can see we have the protein, the DNA ladder, the PCR result, which in this case is showing 2 bands, is what we expected. DNA fragmentation, so a DNA smear. Negative results, so your negative control. And finally, another ladder. Your gel should look something similar to this. When you're ready and done, make sure you take out your gel. You're going to want to remove it. And you're going to want to clean the tray. So just apply a little bit of distilled water. Using Kim wipes, make sure you wipe the tray down, reaching all the corners. It may take some time, but it's important to make sure the tray is clean so that there's no contamination or no damage to the machine. Once you see most of the water has evaporated, you're ready to push the machine back. You can close the tray. And if you're the last one to use the machine, you click the button, you click log out, and you want to click yes. And if you're the last one to use it for the day, you're going to want to click this button again, turn it off, and you should have everything done. That's how you image your DNA gels.